So the first project with Coriolis was way back in 2016. It was called the ACE project, or the Antarctic Circumnavigation Expedition. And a large vessel, a large Russian science vessel was chartered, and it did a circumnavigation of the Antarctic. And a group of scientists bid for projects on that expedition. And I put forward a proposal for an aerobiological project in which we were going to sample the atmosphere all the way around the Antarctic continent to address the hypothesis of whether things are moving in and out, whether they're restricted, or whether they're using the islands as stepping stones to get into the continent. So yeah, it was fairly straightforward actually. It comes in a case, very easy to set up. The requirements are straightforward. I knew what to take. The sample volumes are quite low, so it was easy to take the consumables that I needed for a 90-day expedition. The freezer space wasn't too large, so storing the samples to bring back weren't too big. And the high rate of flow meant that we could sample huge volumes of air in quite a short period of time. Yes, so when we first started, we weren't sure what the best sampling routine would be. So we set up 6 different types of sampling method, of which the Coriolis was just one of them. And we tried to run them for as long as possible each day. So we had a daily sample of the different places we were at around the continent. So to run them for as long as possible, if we could plug it in, we would plug it into the ship and we'd run it for as long as we had the sampling material for. If it was run from batteries, so for example, if we're going ashore on one of the islands, we'd charge up the batteries we had, and we had 3 batteries. So we could run it for about 3 hours in total. We'd run it for that whole period of time. So once we got the samples back, it was about extracting the DNA for genomic analysis. So it's with the liquid Coriolis, we used filter papers. We filtered the water out and then we extracted from the filter papers. For that study, I used different cones for each day because, again, we didn't know what the variability was like. So we didn't know whether we'd introduce issues of contamination. I've since done some tests with some master's students and undergraduates to show that it's perfectly feasible to use the same cone over and over again. So for that particular expedition, we were looking at the taxonomic distribution of bacteria in the air around the Antarctic continent. And we found it to be very variable, and it varied consistently from day to day. So that's interesting in its own right, but we couldn't really link that pattern to any sort of topographical or proximity to land or marine type influences. What we did find, though, that was a very weak correlation with the presence of cyclones and pressure. So there was some indication that weather might have something to do with this kind of diversity that we picked up. But more importantly with that, it showed us that we spent a lot of time and effort monitoring taxonomic distribution of bacteria, when actually maybe we should be thinking about the functional diversity of bacteria. So that led us onto another project in the Arctic, where now we're looking at the functional diversity of the bacteria. And that's much more constrained. It's not quite so variable as the taxonomy. I clean it when I'm back in the UK thoroughly for a new sample run. If between samples in the field, I don't clean it, I just use it because you're sampling air consistently. So the only thing that's going to contaminate it is the day before sample. So if I'm running it continuously, I don't clean it during sampling. But when back in the UK, I clean it according to the instructions in the manual. The challenge is we need as much power as we can get to run the sample as long as we possibly can. So on land, when you're replacing batteries, you need to make sure you run it down to 0 in order to charge it properly. And you've only got as much time as you have batteries available. So I personally had 3 batteries, which meant I could run for about 2 3/4 hours, 3 hours. Then I'd have to charge them all overnight for the next day's sampling. And that constrained the amount of sampling I could do on land to no more than about 2.5, 2 3/4 hours. When we could plug it in, so on board ship or with an electrical generator, sometime I took a generator ashore, particularly when we had helicopter support, you could take a generator ashore, then you could run it off the electrics, and again, you could run it for much longer. Convenience. Comes in a suitcase, you can carry it in your luggage. Fairly robust. You know, I've taken it all around the world. I've taken it on airlines, I've checked it in, I've left it on cargo ships, I've left it on ships while I've not been on the ship. It's been thrown around, still seems to be fairly robust and still works. So I like the size, the weight, the robustness, and the capacity for high sample volumes. What I'm interested in is the distribution the structure of microbial communities and how they influence the environment around them, what role they're playing in Earth system science. So another example, a specific example we're working on at the moment is ice nucleation in atmospheric bacteria over the North Pole. And we're measuring the concentration of ice nucleation genes in bacteria, both in terms of spatial and temporal distribution. So we either go on a transect where we take samples at different points, or we leave a sample in one place and we sample for a long period of time. And from that, we can link the distribution of that functional gene to atmospheric processes. What's happening, where the wind's coming from, what the pressure's doing, what the influence of rain is. Because as the climate changes, we expect the signals that structure the communities to change, and so those functions might change. So we're trying to understand what effects climate change might have on the functioning of the microbial communities. One of the most memorable times using the Coriolis was on Marion Island, which is a South African territory in the Indian Ocean. And we'd come alongside in the ship and we were using helicopters to go ashore. And we were put in a field location away from the station and away from the ship. And while we were sampling, the weather conditions started to get worse. And we got a radio call to say that the helicopters had to come and get us now because it was getting to the window where they couldn't fly anymore, and that they couldn't bring the equipment back. So on the 7th day of the expedition, we faced the prospect of jettisoning all of our equipment in the field and leaving it there while we did the rest of the expedition. But fortunately, one of the pilots, a very skilled and brave pilot, was able to insist on, he tried to pick us up, and he did get us both, and he got all the kit. So that was wonderful. That was wonderful. It saved our project. It really did. Yes. So one of the challenges we have working in the Antarctic is the liquid freezes below zero, and for the ACE cruise, it was never really below zero, so the liquid didn't freeze. But doing the work in the Arctic, we're finding that much more often it is below zero, and it starts to freeze around the intakes and in the sample cone itself. So what I've done is I've got a compact, and I'm using that alongside the micro so that when the liquid freezes, we have a dry sample that doesn't get affected by freezing. And at the moment, last September, we did a transect of the Northwest Passage in northern Canada from Reykjavik to Nome, and we're in the process of working up that data at the moment. And we hope to go back there this year to look at the interannual variation in those patterns that we're seeing, because they are highly variable. And one of the things we noticed last year was that the patterns of microbes in the atmosphere were influenced by things such as the North American wildfires that were happening at the time. So hopefully going back a year later and having a different set of circumstances, we'll be able to do interannual comparisons. It's quite a nice way of getting good data, or lots of data very quickly. And they seem to take to it very well. They like using it, and they generate good results. So I've used them to control some of the experiments that I've been doing in the field.